Hello, welcome to our tutorial video for the second release of the VERTEX simulator. A previous video covers the basics of the VERTEX simulator and introduces you to constructing your own simulations. In this video, we're going to cover two recent additions to VERTEX that are part of this second release. The first is electric field stimulation, which allows users to model the effects of stimulating electrodes on an in vitro slice preparation. The second is synaptic plasticity, both short-term plasticity and spike time dependent plasticity. This diagram shows the process we use to model the effect of an electric field on the neural tissue. We start by constructing a model of the field in the tissue. This involves constructing a 3D model of the tissue using Blender, then using this as the geometry for the finite element model, which we solve using MATLAB's partial differential equation toolbox. One can also use other finite element modeling software such as COMSOL or ANSYS, or can define the field analytically by providing a MATLAB function that returns a value for the electric potential at a given set of coordinates. Having defined the field, and so the electric potential across the tissue, this can be given to VERTEX, which will find and store its value, or values if it is a time-varying field, for the midpoint of each neuron compartment in the model. These will then be used during the simulation to calculate the effect when stimulation is applied. So we will begin by showing how to build a simple electrode geometry using Blender, and then show how to incorporate this into a VERTEX simulation. So to make the tissue geometry in Blender, we want to start by making a cuboid that is roughly the right dimensions for the tissue. So in this case, we're going to go with 30 by 6 by 20, and we will convert these to the correct units in microns when we export. The second thing we want to do is to create the electrode itself. So we can start with a cube again and then adjust this to the correct dimensions. So we're going to go with 0.1 with 6 for the length and 0.1 again for the diameter. Then I want to make a copy of this because we're going to create a bipolar electrode, and then we're going to join these together. And then we're going to use these to create an indent into the tissue cuboid itself. So we can now place the electrode. I want to make sure that this goes further enough into the tissue. And then we're going to use a Boolean operator on the tissue cuboid. So we're going to use a difference operation, and the object we'll use is cube number 2, which is our electrode. So once we've applied that, we can then see that we've got the indentation that we want into our tissue, and those faces there will be the faces that we'll use as the current source in the finite element model. So now we need to reposition the tissue so that it's in the positive quadrant in Blender, so that when we export it, it's in the right coordinate system for the PDE toolbox. So we just need to move it towards the positive quadrant, and we can leave a slight overlap in each dimension with the negative quadrants because we've created a tissue block that's a bit bigger than our actual tissue. So now we just need to export it. So we need to give it a name. We need to click selection only so we don't export the electrode, and we need to scale it up by 100 to get it into the right dimensions. So in MATLAB we can then use the build PDE geometry function, which comes with VERTEX, to load in the STL file that we created in Blender. This will also show us what the STL file looks like, and it will clearly label the faces. This is important because we'll need to use these faces to specify the boundary conditions. We will have three types of boundary conditions. We'll have the non-conducting boundary conditions, which I've called out faces here, and we will have the cathode and we will have the anode. So if we zoom in on the electrode indent into the tissue, we should be able to determine which face belongs to which face number. So this isn't always that easy, but it helps if you zoom right in, and we can see here that F19 belongs to the right one, as does F1 and F3 and F4 and F2. So they all belong to the right electrode. You can see that F5 belongs to the left electrode, F8 belongs to the left electrode, as does F6 and F7. So that just gives you sort of a general idea of how they lay these things out, because it's not too obvious when the faces are this close together. So as I said, we're going to have three different boundary conditions. We're going to have the non-conducting outward faces, we're going to have the cathode, and we're going to have the anode. So to see if we've imported our geometry correctly, we can scroll down and we can use the PDE-plot3D function combined with the built-in VERTEX function for plotting the locations of the somas of our tissue, to make sure that everything is in the right location, and we can see here that it is. So now we want to start to run the simulation itself. So before we do that, we need to set the simulation time. So we're going to run it for 1000 milliseconds, and then we can also set when we want our stimulation to be applied. So we're going to apply it at 600 and 700 milliseconds for half a millisecond of each pulse. So this will be like a paired pulse stimulation. So we'll just let that run. It might take a bit of time. So we'll come back to that when it's finished running. So now that it's finished running, we can plot the results. So we can have a look at the spike raster. So we can see here the effect that we've got from the stimulation, and then we can plot the local field potential, and so we can see we get a field potential response, and we also get an artifact from the stimulation. This diagram shows how synaptic plasticity has been integrated into the synapse class hierarchy. We can see that multiple inheritance is used so that a synapse model can inherit from both a synapse class and a plasticity class. For example, synapse model GEXP-STDP inherits from the conductance-based exponential synapse, but also from the spike-time dependent plasticity class. The synapse model GEXP-MT inherits from the conductance-based exponential synapse class and the Markram and Toddyck's short-term plasticity class. We will now show how these synapse classes can be included in a typical VERTEX simulation. So we will first look at how to add a short-term plasticity model to a VERTEX simulation. So the first thing we need to do to add this is to change the synapse type. So you can see here that we've changed from a regular conductance-based exponential synapse to one that has the Markram and Toddyck's short-term plasticity. So in addition to changing the synapse type itself, we also would need to add in a few extra parameters. We need to add in the time constant for the synapse itself, which is tau s. We need to add in the time constant for facilitation. We also need to add the time constant for the recovery from depression. And we need to add in U, which is the utilization of synaptic resources. So we might also wish to record some additional variables, which allows us to introduce another new feature to VERTEX, which is that you can record from cells specified by their location. So we can specify a location and say we want to record from a certain number of cells closest to this location in particular groups. In this case, we want to record the currents arriving at these cells. So we're going to record from the position 700 by 1500. We're wanting to record 50 of each cell type, and the groups we're interested in are 1 and 3. We're also going to record the short-term plasticity variables, so the variables used by the Markov-Metodix model for the same neurons. So now that we've finished running this simulation, we can have a look at the results. And we can see in the local field potential that we have two responses, one to each of the stimulations. And we can see that the second response is significantly diminished compared to the first, which is known as paired pulse depression. So to find out a bit more about how this paired pulse depression arose, we can have a closer look at the two variables that we recorded, the currents and the synaptic resources. So we can see these if we look inside the result structure, and we can see here how we might investigate them. So we're using the get currents function, and this tells us that the second pulse, when it was applied, there was much greater inhibitory current arriving at the recorded cells, and so this contributed to the depression. And then we can also look at the synaptic resources. So we can see here that the resource available during the first stimulation is greater than during the second. So again, this is also contributing to the paired pulse depression. So now we're going to look at how to include spike time dependent plasticity in a VERTEX simulation. Again, we're going to want to change the synapse type and add in a few extra parameters. As spike time dependent plasticity results in changes to the weights of the network over time, we might want to record these. So we have two ways of doing this. One way is to specify a list of presynaptic neuron IDs, and this will result in us recording the values of all of the synaptic weights coming from these neurons over time for the whole simulation. Another option is to specify time points at which we want to take a snapshot of the weights in the entire network. So here we can do this by specifying the weights array parameter to the recording settings, and here we're specifying it time point 1 and time point 3000. We also need to remember to set the STDP flag in simulation settings to true before running our simulation. So now we can run our simulation and have a look at the results. We can see that when we look at the results that in this simulation we get an oscillation. So in the most superficial layer there's a strong oscillation which entrains the rest of the network. We can now look at those weights that we recorded in snapshots and to show how they've changed over the course of the simulation. There are two ways to plot these. One is to aggregate the change in connection for each group. So we can look at the total change in synaptic weight from the layer 2-3 excitatory cells to layer 2-3 excitatory cells, for example. Another way is that we can look at the change for each individual cell, so how all of its aggregated synapses have changed over the course of the simulation. We'll first look at the group by group weight changes. So we can see here that in this simulation the most of the changes occurred between layer 2-3 excitatory cells and other layer 2-3 excitatory cells and layer 2-3 inhibitory cells. We can take a closer look at this by plotting the change of each individual cell and plotting them according to their location in the network. So we can now see quite clearly here that the more superficial cells have undergone more changes to their synaptic weights. Thank you for listening to this tutorial. If you'd like to know more, then please visit our website, vertexsimulator.org, or have a look at our latest paper, which is included in the description below.